Good morning. I'm going to show part of my PhD thesis. I'm going to talk about the end-Permian mass extinction, and I'm going to bother you again with some geochemistry. But before I head off, I just want to say that geochemistry can also tell us a lot about the ancient biosphere and probably also the role of biota in biogeochemical cycles. And as well learn a lot about the dynamics behind the mass extinction. So, when doing a survey of the literature about the Permian marine ecosystems and the changes across the extinction horizon, I end up with two scenarios that are proposed. One at the left side, where people propose that the trophic pyramid is the top is like taken off, lifted off, and only the base of the trophic pyramid continues to the early Triassic period, so with only primary producers remaining. And then you have the other side, which is also quite famous, the Strangelove Ocean, where it's assumed that the primary producers are like cut off, and that's why the system toppled. But I want to come now with a twist to the story. And I asked myself the question, what could be the role of the primary producers in this mass extinction? As nowadays in the ocean, like these algal blooms that you see here are like major masses of organic carbon that are a major sink as well at the end for carbon, and are an important aspect of the carbon cycle. And for the Permo-Triassic boundary, there is increasing evidence for greenhouse warming as well as increased weathering fluxes, and this could be an integral part of the carbon cycle. So that could make primary producers important in the mass extinction event itself. So we've seen these formulas yesterday as well. Photogenic, oxygenic photosynthesis. I have a somewhat more complicated but still simplified stoichiometric formula. I chose this one with the Redfield ratios because the nutrients are also noted in this formula, and this will come back later in my talk. And then at the flip side, we have the organic carbon respiration. So the breakup of the organic carbon back to CO2, and we have, of course, the aerobic oxidation, and also an important part is the anaerobic respiration, and especially microbial sulfate reduction, also shortly as MSR. Note this acronym, I'm going to use it a lot. And it's said that 50% of the organic carbon reaching the sediment floor is used up by this metabolic pathway. And at the end, we also have this byproduct, which is hydrogen sulfide, which is also implied in many of the extinction scenarios. So my weapon of choice, or my tool that I use, is carbonate-associated sulfate, or also known as CAS, and I measured then the sulfur and oxygen isotope composition. And this is then used as a proxy for the sulfate ion of the ocean water. And the sulfate is used up in this metabolic pathway that I said before, MSR. And this comes along with a large isotopic fractionation, so from the sulfate to the hydrogen sulfide, with up to 35 per mil for the sulfur isotopes and about 4 times smaller for the oxygen isotopes. And it's also important to note that there is a difference in being a sink. For sulfur is only at this end the pyrite burial, and for the oxygen isotopes it's more this reaction, so the metabolic pathway itself. So the material comes from the northwestern part of Iran, the Ali Bashi section and Zal section. They are located in the northwestern part of Iran, close to the Azerbaijani border. so here and here. And there are two main advantages of analyzing these sections for the sulfur isotopes. One is that it's probably an open ocean setting at the time of the Permo-Triassic extinction event. So you can record an open marine signal. And then the second part is the organic carbon content and the pyrite content is low. So the potential for diagenesis upon this sulfur isotopic signal is smaller. So, and that's what we like. So, this is what we like as geochemists. These are our records for the two sections, so the Libashi and the South section, with the Permian and the Triassic. And so we have a very stable sulfur isotopic record for the 2 million years of the Shangxingian, with about only a promille of variation. Similar for the oxygen isotopes, but there is a little bit more spread in the data. It's like about 2 promille. But the interesting feature comes higher up, directly at the extinction boundary. This is the red line here. It's very close also to the administrative boundary of the PTR boundary. And there is a 5 promille oxygen isotope excursion within the sulfate. And at the same time, we see a small negative sulfur isotopic excursion at the same level. Some additional interesting information that could be extracted from this data is we measured also the pyrite fraction of the sediment for sulfur isotopes, and there is a almost constant small offset of only about 15 to 10 promille, which could say that the sulfate concentration of the ocean at the time was maybe low. So, but what do these signals now tell us? So that's where I introduced my box model, a very simplified version of the box model, where the marine organic carbon pool is now visualized as this balloon, and I could then inflate it or deflate it, and this would then drive this reaction, MSR, which is again lined out above here. And what the effect is on the marine sulfate pool for its sulfur and oxygen isotope composition. Although I have to say this is really a simple displaying of this model, because the model also includes fluxes of volcanism and evaporation and weathering and so forth. So now I zoom in to the interval around the extinction, and what we have is this extinction boundary. We have a climate warming event. This is a different record of oxygen isotopes of phosphate measured on comedont material, which shows a negative excursion which can then be related to climate warming. So this could be suggested to drive the model with an increased weathering flux directly at the extinction horizon. And this would then inflate this organic carbon pool, as I said before, and this would also drive again microbial sulfate reduction. And this gives a nice fit for the oxygen isotope composition of Cas, so the sulfate. The line is then the model, and the dots are the data points. But for the sulfur isotope signal, this is a bit different. We need another assumption to create this negative sulfur isotopic excursion. So, why is this negative sulfur isotopic excursion? It seems to be that the pyrite burial flux was not growing proportionally with the MSR. And that could be due to maybe an iron limitation. So, as envisioned in this model of Meyer et al., you have the pre-extinction where there is almost no hydrogen sulfide in the ocean, and then they forced the model with an increased nutrient flux to the ocean, and hydrogen sulfide in the water column expands rapidly. This could mean that your pyrite burial flux would, your formation of pyrite would move from the sediment to the water column, and this could make this regime like more sensitive to iron, and this could also be topped by a change in fluvial regimes where iron coming into the ocean would be reduced. And this could then all produce then this negative sulfur isotopic signal. So to summarize what I've said before, I made these sketches. I hope nobody's going to complain about my drawing skills. But you see at the pre-extinction interval a normal trophic, an ecosystem with the normal trophic structures. And we have pyrite burial within the sediment. Hydrogen sulfide flux is small. But then we go to the extinction event. We have the climate warming. We have maybe an enhanced hydrological cycle. We have maybe the destruction of the vegetation on land, and we get an increased weathering flux with these nutrients, as I showed before. We get a bigger organic carbon pool. So we have more organic carbon coming down to the sediment, to the seafloor, and pyrite formation would now be relocated to the water column. And this would make then the hydrogen sulfide flux also bigger and expanding rapidly. This could then explain our positive oxygen isotopic excursion and the negative excursion at the boundary. And then in the post-extinction early Triassic, we get a return of some fauna in the ocean, but still we have a large weathering flux. But bioturbation within the sediments would have ceased, and it could have relocated the iron, the pyrite formation within the sediment, making the hydrogen sulfide flux to the water column again smaller. And this increased burial of pyrite could then explain this positive sulfur isotopic excursion seen in our record. So to conclude, so maybe in contrast to the Strangelove Ocean, the extinction event is maybe not associated with a dead ocean, but maybe it was still full of life and a large organic carbon flux, and that microbial respiration, like this MSR, could have a big influence on the extinction itself. Thank you.